Okay, so we were talking about the pH and the homeostasis of pH, and we said that the pH is a range between 0 all the way to 14. Right in the middle is 7, which we call it what kind of pH?
Neutral.
Neutral. Anything above 7 is
No, basic.
basic or alkaline. Anything under 7 is
Acidic.
acidic. And acidic means high hydrogen concentration, low pH. Basic or alkaline means the opposite, low H and high OH. Okay, so the pH is inversely proportional to, so it's the other way around, just like we said. It goes up, it's alkaline. It goes down, it's acidic. This is the range from 0 all the way to 14. Obviously, in the middle is neutral, which is 7. And you can move toward alkalinity, toward acidity. If you look at saliva and milk, for example, is a little bit acidic. Tomato and grape is more acidic. Beer, vinegar, wine, pickles is even more acidic. Stomach acidity is about 1. So if you like to eat pickles, if you notice that you feel a little bit of numbness, or your teeth will feel different and your gum will feel different, it's just because of the acidity. It's actually acidic because they add acid to it. On the other hand, egg is alkaline. The ocean water is a little bit alkaline. Bleach is more alkaline. Ammonia, more alkaline. Oven cleaner is one of the most alkaline solutions available. It is between 13 and 14. It's very alkaline. And which one is bad? The acidic or the alkaline?
Acidic.
Alkaline.
Both.
It's both of them. Both of them. Anything that's not normal is bad. The acidic, if you are exposed to something acidic, it will hurt your body. If you are exposed to something alkaline, it will hurt your body. And it's very common to hear little accidents coming to the emergency room because one of the kids picked up oven cleaner or something and they drink it, or the bleach or something like that. This is an accident. So both cases can be harmful. So what's the acid? The acid is a solute that if you add it to the solution, it will add or donate hydrogen. Base is what would remove hydrogen. So you put acid in a solution, it will donate, release hydrogen. You put alkaline or basic in the solution, it's going to take away, it's going to remove acid. So they call it proton donor and proton acceptor, and the proton is hydrogen. Those that donate hydrogen, acidic or proton. Those who remove or take hydrogen or proton, this is called basic. And the difference between strong acid and strong and weak acid versus strong base and weak base. Generally speaking, any one of those that can dissociate easily is considered strong, which is the opposite of what come to our mind. Okay? If I ask you which one is stronger, is the one that dissociate means separate. Okay? So the one that dissociate easily is actually strong. The one that dissociate hard or difficult to dissociate, that will be the weak. And the reason for this is this, that dissociate easily, it can dissociate, releasing its component easily, so it can give more hydrogen, if that's the case. The weak is what will hold on to its components. It's strong to hold on to it, so it's harder to give hydrogen. So this is weak. So if you give more hydrogen, you're strong. If you give less hydrogen, you're weak. Okay, and remember it. Something like, just remember, if you are easy to donate, to give money to poor people or something, you are a strong person, you're a good person. If you are too tight and you don't want to donate anything, you hold on to your money, you are weak. Okay, just something to remember, but you have to know the weak acid versus strong acid, weak base versus the weak base, strong base and weak base. And for the base, it is failure to dissociate in general means weak. Easy to dissociate, whether we're talking about acid or base, is strong. What's the salt? The salt is a solute that dissociates into cation and anion. Any solute, and we understand the difference between solute and solvent, right? What happens when you put a solute in a solvent? What do you call it? Solution, right? You put solute like salt in a solvent like water. You get salted water, which is a solution. Okay? So a solute, something like sodium chloride, table salt. If that become dissociated, means separate to its components, and the result is one positive charge and one negative charge ion, we call it salt, with one exception: water. Water, oxygen, and oxygen is positive, and hydroxide is negative, so why not? This is just an exception, not this one. Everything that when you separate it or when it dissociates give you a positive and a negative ion, you call it a salt, except water. So this is the exception to the rule, even though when you cut it into two ions, it will give you cation and anion, yes, but this is an exception. So sodium chloride, what's the charge for sodium? Is it positive or negative?
It's
Positive. And how about chloride?
negative.
Negative. So is that a salt?
Yes.
Yes. Potassium chloride.
Positive. And
The potassium is positive and chlorine is negative. When you separate them, you get two ions. One is positive and one is negative. One is cation and the other one is anion. Yes, it is. Okay, now the buffer and pH control. What's a buffer? The buffer is compound that contain weak acid and salt, and it can neutralize strong acid or strong base, meaning we have those buffers in our body that's moving around in the blood. If the blood is in the normal range, that's fine. If started to get acidic, this buffer is going to remove the H using the salt part. If it starts to get a little bit alkaline, it's going to the weak acid will release a little bit of its hydrogen. So it's going to buffer. What's buffer means? Mean return it back to normal, balance it. Okay, are we following? So if as long as the pH of the blood is within normal range, it's fine. This is not the case. Usually it starts to move a little bit toward alkalinity, goes up a little bit, goes down a little bit. It's almost impossible to go all the way down, all the way up. It just moves a little bit. What to do? We have these buffers. We have more than one. This is the most important one, sodium bicarb. In humans, this is the most. We have others that we will talk about later on. But for now, sodium bicarb is the most important, or it's a very important buffer that moves around in the blood to buffer our pH. And buffer it means return it back to normal or balance it. Antacids, these are components that neutralize and form salt. If you use Tums or Rolaids or something which is over the counter, like these are medicated. Okay, they are medicated. They contain antacids. There are other types of antacids. If you have acidity or something, and you go to the pharmacy and you get over the counter one of these antacids, whether it's chewable or something that you can use as tablets. Most of it is chewable though. Some of it is even solution, right? I think we all tried something like that before. If you have heartburn or something. So these are antacids. So these are, what's the antacid? It's basic compound. If you feel heartburn, what's the heartburn? Meaning acidity leaving the stomach and going up to the esophagus, so you feel that burning sensation in your esophagus. If you get one of these antacids, it will neutralize it because it's basic. Organic molecules. We said anything to be considered organic, what should it have?
Hydrogen and carbon.
Hydrogen and carbon. It's not only carbon. I'm talking about the in humans. Organic molecules, combined carbon and hydrogen. Don't forget that. Combined carbon and hydrogen. This is the only way to call it organic molecules. Otherwise, you will call carbon dioxide organic molecules, and it's not. Right? It's not only carbon. It's both of them together. And they have to be covalently bonded. Usually oxygen. So practically speaking, I'm not saying if you have carbon and hydrogen only, it's not organic. It is. But practically speaking, in our body, most of it, beside the hydrogen and chloride, usually, also have oxygen. And there is always a functional group beside those. Of course, it's not hydrogen and carbon. This is not a real organic compound. But the rule apply, yes. Anytime you see a compound that contains carbon and hydrogen, just call it organic. But to be practical, most of the time it contains oxygen. Most of the time it will contain functional group that determine the chemistry. And the organic molecules that we know is carbohydrate, lipids. What's the other name for the lipids? Fat. Proteins or amino acids and nucleic acids. So the four nutritional or the four organic compounds that we know: carbohydrate, fat or lipids, protein, nucleic acid. I want you just to know carboxylic acid, just this. Carboxylic acid is COOH, amino group is NH2, hydroxyl group is OH, phosphate is PO4. Anything else is, at this point, is not something that we will focus on. So just remember, when you see COOH, it means carboxylic. NH2, amino group. OH is hydroxyl group, and PO4, that will be the phosphate group. Now the carbohydrate. Carbohydrate, is it organic or not organic?
Organic.
It's organic, so it does have carbon and hydrogen, and it will have oxygen too. Most of the organic that we will talk about will have oxygen, and the ratio is 1:1. What's 1:1? Carbon to hydrogen to oxygen. It has to be in this ratio. And instead of memorizing the formulas, you don't have to memorize, just remember the ratio. So when I give you something, C3H6O3, is it carbohydrate or not?
Yes.
Yes. C5H10O5.
Yes.
Yes.
C6H11O6.
No.
No. It has to be 1:1. So you don't have to memorize the formulas, just look at it. It's the ratio 1:1. Yes, carbohydrate. That's it. Okay? Carbohydrate can be monosaccharide, disaccharide, or polysaccharide. So if it is a simple sugar, 1, and this is the building unit for carbohydrate, mono means 1, di obviously means 2, poly means many, which is more than 2, 3 or more. Okay, so monosaccharide is a simple sugar, and this is the building unit of carbohydrate. Disaccharide is 2, polysaccharide is more than 2. Monosaccharides will have from 3 to 7 carbon atoms. So if I told you, listen to this, if I told you 2 carbon, 4 hydrogen and 2 oxygen. Is it carbohydrate? It does obey the ratio. Is it carbohydrate? Is it monosaccharide? It is not a monosaccharide. The minimum is 3. Okay? Less than that, you don't call it monosaccharide. It can be another thing related to carbohydrate, maybe, but it's not a monosaccharide. We don't have monosaccharide that's less than 3. So it cannot be 1 to 1, 2, 4, 2. No. Anything from 3 to 7. 8, 16, 8, no. 3 to 7. And the examples are glucose, fructose, and galactose, and you have to remember these. Okay? If I ask you which one of the following is a monosaccharide, for example, and I give you fructose, maltose, starch, you need to tell me that it's fructose. Okay? So you have to remember these names. Glucose, fructose, and galactose, these are monosaccharides. Disaccharides are 2 monosaccharides condensed. Do you remember these types of reactions? Do you remember the anabolic versus catabolic, the building versus breaking down? Do you remember this or not?
Yes.
Okay, so you get one monosaccharide, you get another monosaccharide, they will condense together to build a disaccharide. So this is synthesis reaction. And the synthesis reaction, if you still remember, we called it dehydration. Do you remember this? What's a dehydration? What will happen before this monosaccharide and this monosaccharide come together and build a disaccharide? What they have to do?
They lose their water.
Yes, they lose the water, both of them. One of them will lose hydrogen and the other one will lose
Oxygen.
not oxygen. Hydroxide, yes. So if I'm a monosaccharide, you're a monosaccharide, can we make disaccharide together? Yes. Lose H first and I will lose OH, or the opposite. So this will be released and then we can bind together by a covalent bond to make a disaccharide, which is called dehydration synthesis. What's the synthesis? We synthesized, in this case, we synthesized disaccharide. What's dehydration? Without hydration. What's hydration?
You get water.
Water. So dehydration to get rid of water. I lose H, you lose OH. Both of them together will make water. So we ended up uniting together and releasing water. Dehydration synthesis. And this is, we will talk about this a lot. Every time organic compounds come together, it's always dehydration synthesis. So don't forget this. Examples, sucrose and maltose. Sucrose is the table sugar, the regular sugar. Okay? Maltose is the malt sugar. And those two are disaccharide. And the maltose is 2 glucose units. So this is disaccharide. There is one more. I don't know why they didn't put it here, but this is something you need to know, which is lactose. It's a disaccharide. So sucrose, maltose, lactose. Lactose is the milk sugar, the milk disaccharide. Maltose is the malt disaccharide. Sucrose is the table sugar. This is the common name for these structures, 3. So I want you to know 3 from each one of those. Polysaccharides. Same thing. You get one monosaccharide, add another monosaccharide, dehydration synthesis, and add another monosaccharide, and add another monosaccharide. So dehydration, water after water after water after water. So you're condensing, and at the end you're making a polysaccharide, which is glycogen, starch, and cellulose. Glycogen is the storage form of polysaccharide in human and animals. Starch is the storage form of polysaccharide in plants. Cellulose is the storage form of polysaccharide in part of the plant, which is the outer cover that's not eatable, right? Can you eat the trunk itself? Can you eat the leaves? You can't eat the leaves. You can eat something that stores starch, like the fruits, like the vegetables, right? Animals can, right? We see animals eating the leaves of any tree, right? Why? Because the leaves have cellulose. Cellulose is a polysaccharide. We don't have that enzyme. Animals does. They have an enzyme that can break cellulose and they get the components out of it, which is glycogen. So cellulose is a polysaccharide, consists of glucose. Starch, same thing. Glycogen, same thing. The three of them are many units of glucose. Okay? But glycogen is in humans and animals. Starch is in plants, which is the edible form. Cellulose is a non-edible form in humans of polysaccharide. And again, you have Nine's name to know. Look at this and tell me, is it carbohydrate or not? Count. How many carbons?
5, 6,
6. How many hydrogen?
6, 7.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 hydrogen. How many oxygen? 1, 2, 3, 4, 5, 6. Is that a carbohydrate?
1 to 1, yeah.
1 to 1? Yes. So do you have to memorize this? No. If I show you any formula, just look at it. Is it 1 to 1? It's carbohydrate. Okay? So this is the dehydration sense. I'm not going to ask you about any formulas. Okay? Just look at it to understand what I was talking about. This is a fructose. And this is glucose. Glucose will lose the OH. Fructose will lose the H. What is this together? Water. So this is dehydration synthesis. Right? Dehydration synthesis. And this will give you the sucrose. So what is the sucrose again? The two components that make the sucrose that I just mentioned? Sucrose.
Two mono.
It's two monosaccharides, yes. But what are these? Glucose and fructose. The sucrose is the table sugar. It's a disaccharide. Okay, what are the two saccharides? What are the two monosaccharides? Glucose and fructose. Maltose is is 2 units of glucose. How about starch, glycogen, and then cellulose? What are the components? What type of monosaccharide? What are the components?
Units of glycogen.
Of glucose.
Oh, glucose.
Units of glucose. Are you still sleeping? The three polysaccharides that we know are just glucose. Multiple units of glucose, that's it. Maltose, 2 units of glucose. The table sugar that we use it for tea or for coffee or anything else, this is 1 sucrose and 1 glucose, okay? How about the other way around? I give you a sucrose and I ask you to break it down into the two components. What are the two components? Glucose and fructose. What do you call this reaction then? Which is the opposite of dehydration?
Hydration.
Hydrolysis. It's close to hydration. It's called hydrolysis. So you build dehydration. You break down hydrolysis. What's hydrolysis? Just the same thing. I am sucrose and she's glucose. Can we make disaccharide together? I lose hydrogen, she lose OH. Okay, now we are disaccharide. All right? Can we break down? Give me back my hydrogen. Give her back her OH so we can break down. So you're adding water, which is the opposite, right? This is called hydrolysis. Dehydration is the opposite of hydrolysis. Did you get this? If you wanted to put two together, take hydrogen from one, Take OH from one, this is together water. So this is called dehydration. You removed water. I want to do the opposite. You want to break it down? Yes, you can't. Why? Because I lost my hydrogen. I'm not going anywhere before getting back my hydrogen. And she's not going anywhere before getting back her OH. Give us back the water. So this is hydrolysis. Hydrolysis means lysis using water, breakdown using water. So this is hydrolysis. And that will give you back the glucose and fructose. This is the glycogen. All of these units are just glucose. Okay? Glucose, attach another glucose, dehydration synthesis. Dehydration synthesis, another glucose. And then you make a branch. Glucose, branch. So it's branching polysaccharide. And of course I don't want you to memorize this or remember this or something. Just know, have an idea about it. It's a branching polysaccharide consists of glucose. Okay? Right. So monosaccharide, disaccharide, polysaccharide, 3 names from each one of those, of the 3 that you need to remember. Now the lipids. The lipids is hydrophobic. What's phobic means? Fear or hate. So what's hydro? Water. So they hate water, meaning they don't dissolve in water. This is fat, oil, and wax. And this is mostly carbon and hydrogen. We're not talking about oxygen now, not much. Examples, fatty acids, eicosanoids, glyceride, steroids, phospholipids, and glycolipids. We will have some more details about this later on, but you need to remember that these are all lipids. Fatty acid, eicosanoid, glyceride, steroid, phospholipids, and glycolipids. We will discuss the details of that later on. Fatty acid is a long chain of carbon and hydrogen, but it will end with carboxylic acid. This is what I just told you to remember. You see this, COOH, carboxylic acid. So you don't memorize. You see carbon, hydrogen, hydrogen, carbon, hydrogen, carbon, and at the end you see COOH, know that this is lipid. Do you get how to know the lipid now? How to know the sugar? 1 to 1. That's it. Sugar. Just by looking at it. You don't memorize. Just count. Okay, and usually I don't give you that much formula. I get you something like C5, H10O5, for example. Is it 1 to 1? Easy? Yes. Carbohydrate. If you see a molecule that have carbon and hydrogen, and at the end you see the carboxylic acid group, that's it. This is fat. What type of fat? This is too much details. Just when you see this at the end, know that this is fat. And the fatty acids, the fat in general is non-polar, and we talked about polar and non-polar before. So non-polar is hydrophobic, and the only thing that's polar is the carboxylic group. Fatty acids can be saturated and can be unsaturated. Which one is better for our health? Saturated. What should you eat? What should you use? Saturated or unsaturated? Which one is healthy?
Unsaturated.
Unsaturated is. Saturated is the bad one. Okay? So we try to avoid saturated fat. And what do you mean by saturated versus unsaturated? Saturated means each atom have enough atoms reacting to it, so there is no need to make double bonds or covalent bonds. Saturated, you have enough hydrogen, so you don't need to make a covalent bond. Okay? So, meaning, if I'm a carbon, okay, and I have 4 on my outer shell, so I need another 4. If you give me 4 hydrogen, I'm fine. I'm satisfied. I'm saturated. Okay? You give me 4. That's what I need. If you don't give me 4, I'm not saturated. And I have to find another way. So let's say I am carbon and you give me 4 hydrogen. I'm okay, I'm fine, I'm saturated. You give me enough hydrogen. That's it. You only give me 2 or 3. I'm not saturated. You give her 3. She's not saturated. What should we do? How about we make a double bond together? I share with you. So we will make a double bond. Okay? So this is not saturated. When you have a double bond, you are forced to. So atoms in general, they don't like to make a double bond unless they are forced to. So if you give it enough hydrogen to be saturated, it will be fine. If you don't, the other way around is, let's get 2 carbon molecules that make a double bond that will not be saturated, unsaturated. Okay? If you have 1 double bond, that's called mono unsaturated. So it's unsaturated. Unsaturated, you mean you have a double bond? Yes. If you have 1 double bond, you call it mono unsaturated. If you have more, 2 or more, you call it polyunsaturated. Did you get this? So the saturation is you have enough hydrogen to saturate you, to be satisfied. You are satisfied. You are saturated with hydrogen. Okay? And for your information, just from the nutritional point of view, anytime you see any food that say hydrogenated something, avoid it. Why? Because what's the hydrogenation in general? Anything that's not hydrogenated, that have the double bond, will be in a liquid form. Okay? Like oil, for example. So they take the oil, they force hydrogen on it. This is called hydrogenated. So when you add hydrogen, it will become saturated and it will become solid. And this is some of the margarine that we see. You see it's solid, it looks nice. They add color to it, so it looks very nice. Okay? What is this? It was an oil. It was oil. And they added hydrogen to it. So the unsaturated oil, which was good, you added hydrogen to it. Now it became saturated, and now it became bad. And they are just doing that to make it solid. Did you get this point? The unsaturated is healthy. The saturated is unhealthy. The unsaturated is usually oil, or not oil, I mean liquid, and the saturated is solid. And again, as I told you, if you see hydrogenated something, hydrogenated olive oil, hydrogenated vegetable oil, something like that, know that this was an oil and they change it to replace the margarine. It looks like margarine, looks good and everything, but it was an oil, hydrogenated, so it's not good. Not healthy. So here's an example. Do you see from this part? Do you see oxygen? You see any oxygen here? Yes. No. Is it all hydrogen and carbon?
Yes.
And what is this? COOH. What do you call this? Carboxylic group. So what is this? Fat? Yes. How to recognize, again, how to recognize the sugar? 1, 2, 1. Carbon, hydrogen, oxygen. How to recognize the fat? Carbon and hydrogen, and at the end, carboxylic group. Okay? Did I ask you to memorize this? No. Just look at it. Is it carbon and hydrogen? Yes. Does it have COOH at the end? Yes. That's it. So you know that this is fat. Which one? This is not the issue. Just know if it is fat or carbohydrate or protein. We will talk about the protein. This is saturated and this is unsaturated. What's the difference between this and that? What did you notice? Look at it and tell me. This one contains double bond and this one does not. Okay, so this is unsaturated, which is good. This is, you added hydrogen, so you break down this. So the carbon become saturated which is bad. Okay. Eicosanoids, this is one of the lipids. They are derived from fatty acid. So you get the fatty acid. That's called arachidonic acid. You change it to eicosanoids. Leukotriene is an example. Prostaglandin is an example. Leukotriene is important for immune system. We don't need to know the details now. Prostaglandin is just a local hormone. So these are fats that we need to have an idea about. Okay, this is another structure of the fat. Glycerides. What is the glycerides? Glyceride. Two parts. Glyceride. Glyc means glycerol. Ride means fatty acid. Remember this. Glyceride. Glyc is glycerol. Ride is fatty acid. So when I say glycerol, know that this is, glyceride means glycerol plus fatty acid. If I told you this is triglyceride, tri means three.
Glycerol,
Glycerol, right?
fatty acid.
Fatty acid. This is a glycerol and 3 fatty acids. Okay? Got it? Triglyceride. What does it mean? Glycer means glycerol. Ride means fatty acid. Triglyceride means 3 fatty acids and one glycerol. Did you get this? This is a triglyceride. How about the diglyceride? Two what?
Glyceride. No, 2 fatty acids.
2 fatty acids and 1
Glyceride.
glycerol. Okay? Always try to remember the prefixes and suffixes that I'm talking about, because it will help you every time we mention it again. You will use it over and over. So the triglyceride is basically, this is the fat under our skin, right? You gain fat, you lose fat. We're talking about basically triglycerides. Okay? So this is the most abundant fat in our body, triglycerides. So it's used for energy, insulation, and so on. So this is just the fat under our skin. This is a triglyceride. This is glycerol. How many fatty acids? 1, 2, 3. 3 fatty acids. So this is triglycerol. This is how it looks like. So you add glycerol, fatty acid, add another fatty acid, add another fatty acid. You end up making triglycerides. And let me ask you this and see if you are following or not. Is this anabolic or catabolic? From this to this. This is 1 glycerol, 3 fatty acids. Each one of these is separated. And you end up making this. Is it anabolic or catabolic? Anabolic? Yes. What kind of anabolic? Is it dehydration or hydrolysis? Dehydration. Are you following or not? Are you following? Dehydration? Yes. In all organic molecules that we will talk about, I'm talking about general rules here. When you understand the rule, apply it for everything. It will always work. Okay? Anytime you do anabolic reaction in organic molecules, it's dehydration synthesis. Anytime you break down an organic molecule, which is catabolic, it's hydrolysis. Can you remember this? Dehydration synthesis versus hydrolysis. Dehydration synthesis, anabolic, versus hydrolysis, which is catabolic. Can you remember this? Dehydration synthesis versus hydrolysis. Dehydration synthesis is anabolic, building. Hydrolysis, catabolic, breaking down. Right? Is that only here? No, it's in everything.
Wait, I thought it was the opposite. I thought when you break down, it's hydrolysis.
Yes, did I say the other way around?
Yeah.
Okay, I'm sorry. Take it back. When you build, it's anabolic, it's dehydration synthesis. Catabolic, it's hydrolysis. Okay? So how about when we talk about the amino acids? You add one amino acid to another amino acid. Is it anabolic or catabolic?
Anabolic.
Anabolic. Is it dehydration or hydrolysis? Dehydration. How about glucose and glucose and glucose? Is it anabolic or catabolic?
Anabolic.
Anabolic. Is it dehydration or hydrolysis?
Hydrolysis.
How about if I get sucrose which is 2 glucose held together, and I break it down. What do you call this? Catabolic? Is it hydrolysis or dehydration? Hydrolysis. Did you get the rule?
Yeah.
For everything. Anything, any organic molecule, same thing. Steroids, this is one more of the lipids, but they contain rings. This is the difference. When you see a ring, know that this is a steroid. Okay, besides a functional group. It can be cholesterol, estrogen, testosterone, corticosteroids, calcitriol, and bile salts. These are all steroids that we need to know. Cholesterol is part of the plasma membrane, and we will cover next chapter. We will talk about the details of the cell, including the plasma membrane, and you will learn more about it. But for now, know that cholesterol is part of the plasma membrane. So some of us will think that cholesterol is bad. I shouldn't eat cholesterol. No, that's not true. You actually do need cholesterol. You need it for the cell membrane. If you don't eat cholesterol, your cells will die, because it's an essential component of the cell membrane. So should I avoid cholesterol? No. You should get enough cholesterol, not above your needs. This is, so anything in general, anything that you think it's not good, there is nothing that's good or bad. It's not like this. Anything within the normal limit is good. Anything beyond, under, or above the normal limit is bad, irrespective of what we think. Cholesterol is bad, we don't say bad. Above normal cholesterol is bad, under normal cholesterol is also bad. So what do you need? Enough amount of everything. Estrogen and testosterone. What's the estrogen? Female hormone. Testosterone. So this is steroid. Corticosteroid. This is like cortisone. So this is lipid. Calcitriol. This is a hormone released from the kidney. And bile salts. All of these are steroids. So it looks like this. Again, the chemical formula is too much details. Just because they have ring, this is steroids. Phospholipids and glycolipids. Cut the word into 2 pieces and tell me. Phospholipids, what do you think?
Lipid.
Lipid plus phosphate. Yes. Phospholipid. Phosphate and lipid. That's it. Okay? Phospholipid. How about glycolipids? Glycolipids. It's glyco, which means sugar, and lipid. So this is sugar and lipid together. Glycolipid. Just as a general rule, AP1, AP2, anything. Always cut the word into pieces, and you will get it. Glycolipids. Glyco means sugar. Lipids is lipid, fat. So what's a glycolipid? It is sugar and fat. Phospholipid. Phospholipid. Phospho, which is phosphate, and lipid. Okay? Phospholipid. Diglyceride. We talked about the diglyceride. Diglyceride. What's di? Glycer.
Sugar.
Glyco is sugar. Glycer. Glycerol. And right? Fatty acid. So what's a diglyceride? 2 fatty acids and 1 glycerol. Okay. So when you add the diglyceride to phosphate, that will be phospholipid. If you add it to sugar, that will be the glycolipid. Phospholipid, these 2 components are parts of the plasma membrane. Glycolipid and phospholipid. We mentioned cholesterol already. You will see the details next chapter. So this is a phospholipid. The phosphate group is this yellow. This is the non-lipid component, and glycerol and 2 fatty acids. So diglyceride, 2 fatty acids and 1 glycerol, adding phosphate and non-lipid. Again, I'm not going to ask you about the chemical formulas, okay? Except the part that I'm talking about. I'm not going to ask you like to draw something or tell me what is this, like structures like these. I will ask you, however, to tell me if this is sugar or not. The simple thing, okay? 1 to 1. Is it lipid or not? Is it CH and COH at the end? That's it, okay? We will know how to recognize the proteins too. So this is the phospholipid and glycolipid. It looks like this, just for your information. And this is a cell. This is the membrane outside covering the cell. Proteins. This is the most abundant and important organic molecule. What abundant mean?
A lot of.
A lot of, or the most available. And it contains carbon, hydrogen, oxygen, and
Nitrogen.
nitrogen. So when you see nitrogen, know that this is protein. Do you know now how to recognize carbohydrate from fat from protein? Just by looking at it. Okay? This is how to tell. And the basic building block is 20 amino acids, at least. So it's 15 protein, 15 amino acids? No. 20 is protein, 50 is protein, 200 amino acids is protein, 19 is not a protein. What do you call it? It's called polypeptide. Isn't the protein, is it polypeptide? Yes, it's a polypeptide, but 20 or more. Are we following? You put amino acids. So what's the building unit of proteins? Amino acids. What's the building unit of fat? Fat. Fatty acid. Fatty acid and glycerol, if it's triglyceride. But fatty acid. What is the building unit of carbohydrate? Sugar. What type of sugar? The simplest unit.
Like glucose.
Glucose is one of them. No, sucrose is two. Glucose,
Monosaccharide.
monosaccharide. Yes. One more time. What's the building unit of carbohydrate? Monosaccharide. What's the building unit of fat? Fatty acid. What's the building unit of protein? Amino acid. So you put amino acid plus amino acid plus amino acid plus amino acid. You call it polypeptide. Until you get to 20. You call it a polypeptide, but you call it protein. Okay? Did you understand what I'm talking about? Let's say we're talking about bricks in the wall. Okay? I got 15 bricks. Can you call it a wall? No. You have to make a whole wall in order to call it a wall. But it is part of, yeah, but you don't call it a wall. So you put amino acids beside each other, you only call it polypeptide. What if it adds up to 20 or more? Now you can call it protein. Right? So 15 is a polypeptide, 50 is a polypeptide, 100 is a polypeptide. But under 20, you only call it polypeptide. Above, from 20 or more, it's a polypeptide that is protein. So in order to qualify the polypeptide to be a protein, it has to be 20 amino acids or more. What's the function of the protein? Support, like structure protein in our body, like muscles, for example. Movement, muscle. Contractile protein, which is muscle. Transport, we will talk about this. It's a carrier that carry things in the blood. Buffer, we talked about the buffer before, which regulate the pH. There is some buffers that are protein. We will add the buffers together later on. Metabolic regulation, enzymes are proteins. Hormones are proteins. Antibodies are proteins. Okay? Hormones, enzymes, antibodies, one of the buffers, this is all protein. All right? Hormones, antibodies, enzymes, buffers, proteins. So what's a protein? It's a long chain of amino acid that's 20 or more. What's the amino acid? It's a central carbon plus hydrogen plus amino group. And this amino group, which contains the nitrogen, is your way to know that this is a protein. When you see the NH2. Okay? You know that this is amino acid or protein, which is the same thing. Okay? This is, you have carboxylic acid on the other hand. That might confuse us with what else have carboxylic group? Huh? Fat. So can you be confused between the fat and the proteins?
Yeah.
You shouldn't. Why?
There's nitrogen.
Because number one, yes, you do have carboxylic, but you have NH2. Do you have NH2 in the fat?
No.
Of course not. This is one. Number two, do you have oxygen in the middle of the structure of the fat? It's only CH, 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 CH. You don't have. In this you have. But it's enough to say, if you have carboxylic acid group without NH2, this is fat. If you have carboxylic group and NH2, which is amino group, this is protein. Can you now recognize a carbohydrate and fat and protein just by looking at it? Okay? If you find carbon, hydrogen, oxygen, no carboxylic or NH2, and 1 to 1, this is carbohydrate. Okay? You see COOH, look for NH2. It's not there. Just carbon and hydrogen and carboxylic. This is fat. You have the carboxylic plus NH2, this is protein. So there is an amino group, carboxylic group, and R group. And R group is the additional group. So it looks like this. This is the central carbon. This is the NH2 from one side. This is the COH from other side. You have hydrogen and you have the R. And the R group is different from protein or amino acid to the other. Dehydration synthesis. We talked about this. When you add any two organic molecules and you're trying to make a larger molecule, it's anabolism and it's dehydration. Always remember this. And here is where the name polypeptide came from. This is one amino acid. And this is another amino acid. I'm going to attach them together. What I should lose now? Water? Yes. They are held together now. This bond is a special bond that's called peptide. So here is amino acid, another amino acid, peptide in between, amino acid, another amino acid, peptide, 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 peptide. What do you call many peptides?
Polypeptide.
Polypeptide. This is where the name came from. Okay, same thing, dehydration synthesis versus hydrolysis. These are examples of amino acid, glycine and alanine. These are, both of them are amino acids. You wanted to put them together. This dehydration synthesis, can I break it down? Yes, it's hydrolysis. So it goes either way. And this is the peptide bond, which is called, and this is called, the bond that's called peptide bond. Now the protein structure. The protein structure, in order to call it a protein, the protein is a complex. It's not only that you get some amino acids and attach them and you call it a protein. No, it's not. It's not like this. It's not as simple as this. Okay? So in order to make a protein, you don't just add some amino acids. It doesn't go like this. There is a primary structure. Okay? Like if you look at the wall, for example, not this, but the regular wall that's made of bricks. I put one brick after one, after one, after one, after one. This is the primary structure. Okay? Secondary structure, I'm going to bend. So amino acid, amino acid, amino acid, amino acid. This is a primary structure. When I start to fold it, like you make the wall bending, okay? This, bend, bend. Now this is the secondary structure. Tertiary structure, which is the third one, is this secondary structure. You're going to fold it. Okay? So like the whole room. This is a tertiary structure. Quaternary structure is the final shape, which is this room plus the next room plus the bathroom plus the hallway. This is all together the quaternary or the final structure. So primary structure, just some amino acids. That's it. Just blocks of amino acids. Start to make it a spiral or start to bend. Secondary. Fold it. That would be tertiary. Put it in the final shape. That would be quaternary. And primary, secondary, tertiary, quaternary means 1, 2, 3, 4. Okay? Primary, secondary, tertiary, quaternary. So what is this? Primary. You start to fold it. What do you call this?
Secondary.
Secondary. And in order to keep it in this position, you need to make some bonds. Otherwise, if you didn't make the bonds, you fold it, let it go, it will go to the primary structure again. So when you fold it, you need to make some bonds to keep it in place. And this is called the secondary structure or the alpha helix. This is when you bend it. See, this, look at this part only. What is this?
That's for primary.
This is primary. What is this?
Secondary.
Secondary. And now you get the secondary and you bend it, fold it. You call it tertiary. The final shape is tertiary plus tertiary plus tertiary plus tertiary. Okay? So this is like some bricks. You fold it. So you're making some walls. You bend it, making the whole room. You have the bedroom, the bathroom, the living room, all together like a whole house. That will be the complete protein. Primary, secondary, tertiary, quaternary, and quaternary has multiple tertiaries. So tertiary is like a room. Okay? Protein can be, shape of protein can be fibrous, can be globular, looks like globule. These are different shapes, and this will, the function will depend on this. So something like keratin and collagen, this is fibrous, and this is quaternary structure. This is the final structure, basically. Okay, now the enzyme function. We talked about enzymes as catalysts. What's the function of the enzyme? What does it make with the reaction? The reaction is happening anyway. And when you use an enzyme, what will happen to the reaction? Less energy and faster, right? Do you remember the hydraulic machine when I told you your car, you have a flat tire in the middle of the road and nobody's assisting you and you spend time and effort to replace the car, but if I give you the hydraulic machine, it will take a couple of minutes and no much energy, right? No much effort. So it's just like the hydraulic machine. It reduces the energy needed and the reactions goes faster. Okay? The enzyme itself should not change by the reaction. It just helps the reaction. The reaction is over, it go and help another structure, another reaction. Enzymes are specific, limited, and regulated. What does it mean? It means specific. Do you want me to catalyze reaction? Yes. Which reaction? Water and hydrogen. We wanted to make water. Oxygen and hydrogen. Well, we wanted to make water. Yeah, that's okay. I can do that. How about carbon dioxide and water to make carbonic acid? No, that's not. I'm not specified in this. Okay? So they have specificity. Okay? You use the hydraulic machine to change tire. Can you use it to fix the engine? No. It's specific for the tires. There's another one that can work with the engine. There is a third one that can work to do the glass, for example. So each one is specific. Not all enzymes help all reactions. They are specific. Limited, meaning they can be saturated. Okay, I have 5 cars that are broken down, and I have 2 hydraulic machines only. This is beyond, I'm saturated. So you have to use them, and after you finish, you can go to the next one. So this is limited, and it's regulated, meaning the function itself, the cell can tell the enzymes what to do. It's regulated. So this is how it goes. This is the enzyme. We'll have 2 spots. One of them is for the first substrate, second substrate. They come together, react, make a product, release. And the idea is less energy and faster. There are other things that help the enzymes. Helps the enzyme itself. Cofactor, coenzyme. Cofactor is a factor. Co means together. So this is a factor that work together with the enzyme. It's an iron molecule. And when it binds to the enzyme, it will help the enzyme do the work. Coenzyme is a cofactor, a type of cofactor, but it's not protein. So cofactor is anything. Cofactor is anything that helps the enzyme. Coenzyme is a cofactor, but not protein. Isozymes, two enzymes catalyze the same reaction. Okay? So you can have two enzymes working together in the same reaction. Two hydraulic machines are used together at the same time. Just do this, you're done. So you can use both of them working on the same reaction. Heat, pH, denaturate proteins. What do you mean denaturate? What happens if you get the egg, the raw egg, when you break it down, isn't it liquid, the white? Isn't it liquid? Okay, expose it to heat a little bit. What will happen? Solid and white. Is that the nature of the egg, or the nature changed? Changed. It was liquid, colorless. It becomes solid, white. So it's denatured. It's the nature changed. So when you expose it, how about this? Another thing. You get the same egg, and the white part, which is colorless, transparent, liquid, and get some acid from your stomach and put it on it. It will happen the same thing. You didn't see that? I saw that in the lab. Get some eggs, put some acid on it, same thing. Just like you exposed it to heat. So the point here is both changing pH and heat denature, changing the nature. So it will lose the shape and function. Can you return back the egg after it becomes solid? Can you return it back to the original status? No, it's changed forever, right? So it will lose the function and the shape. Glycoprotein and proteoglycan. Cut this into two pieces and cut this into two pieces. It's the same, but there is a difference actually. But cut this into two pieces. What is this?
Glyco, sugar.
Glyco, which is sugar, and this is protein. Sugar, protein. Proteoglycan. Like. What is it?
Protein
and
sugar.
It's the same thing, isn't it? What's the difference then? Both of them are protein and carbohydrate. There is a difference. Glycoprotein is large protein and small carbohydrate. Proteoglycan is the opposite. Okay? So glycoprotein, the second part is a large part. Proteoglycan. Get the difference? Glycoprotein. Proteoglycan. If the protein is larger and the glucose is small, the sugar is small unit of sugar, and large protein, you call it glycoprotein. If the glucose is, the carbohydrate is large and the protein is small, you call it proteoglycan. You get the difference? So it's the second part. The second part is large. Glycoprotein, protein large. Proteoglycan, the sugar is large. And basically, the glycoprotein is most of what we know. This is the most common one. Enzymes, hormones, antibodies, everything. And proteoglycan is the only one that's special. It's for viscosity, to make things a little bit viscous. Like if it is liquid, it will become thicker. This is the only difference. So this is proteoglycans. Nucleic acid. You have until 9:30, right? 9:30?
Yeah.
Okay. Nucleic acid. Nucleic acid is DNA or RNA. Both of them are nucleic acid. So far we covered 3 out of the 4 organic compounds. What are the organic compounds that we know?
Fatty acid.
Fat, protein, carbohydrate, and nucleic acid. We covered the other 3, right? Carbohydrate, fat, proteins, and this is the nucleic acid. Nucleic acid can be DNA or RNA. What's the DNA stand for? Deoxyribonucleic nucleic acid. Deoxy. What's D?
No.
No. Oxy.
Oxygen.
Ribo means ribose. It's a sugar. Nucleic acid. So let's take it this way. It's a nucleic acid
No.
that have a ribose that you removed oxygen. Okay? Can you get it this way? Always apply this. Trust me. It will always work. Cut the word into pieces. It will make it a lot easier. Deoxyribonucleic acid. It's a nucleic acid, isn't it? Nucleic acid. And this is deoxyribo. Means it is the sugar ribose, but you remove the oxygen. Ribose with removed oxygen. Deoxyribose. How about if you didn't remove the oxy? Ribonucleic acid. This is a nucleic acid that contains the sugar ribose, but you didn't remove the oxygen. You get the difference? RNA is ribonucleic acid. Nucleic acid that has the ribose as it is. The sugar ribose as it is. If you get this sugar ribose and remove oxygen out of it, you call it deoxyribonucleic acid. So both of them are nucleic acid. Both of them have the sugar ribose, but one of them oxygen removed from it. Can you understand it this way? Try to apply this, and trust me, it will always work. 233, 234, anything in the medical field or healthcare field, anything in the medical terminology, just apply this. It will always work. I've been teaching a lot of things, not only physiology and anatomy, microbiology and immunology and nutrition and pathophysiology and a lot of things. It's always the same rule in the medical terminology. Just cut it into pieces, apply the prefixes and suffixes, it will work. And it will make it a lot easier, because next time maybe you're using D alone or deoxy alone, right? So understand where the word came from, and it will make a lot more sense. So DNA, what's the function of the DNA? It will determine the inherited characters. So the characters inheritance, how tall you are, what's your function, color, everything, all characters of the humans are kept within the DNA, not the RNA. So the big boss is DNA. This is the one that determines. It directs the protein the protein synthesis. This is what tells you what type of protein you make and when to make it. Enzyme production. You need enzyme. I will tell you which enzyme to produce. This is the manager that determines everything, the DNA. So I will tell you which enzyme to produce and when to produce it. And metabolism, I will tell you. You are exercising, you need more metabolism. I will tell you to improve or increase your metabolism. You're cooling down, you're relaxing. I will tell you to downgrade or relax or decrease the metabolism. So it's controlling everything. How about the RNA? The RNA is just control the intermediate steps, because this is just the helper for the DNA. Okay? So the DNA is doing all of this, right? The DNA wanted to make protein. I will ask the RNA to help me in doing the proteins. Okay? So it's helping in one of the functions of DNA, which is protein synthesis. All right? The structure, generally speaking, of the nucleic acid, whether it's DNA or RNA, it is nucleotides. So this is the number 4. What was the building unit of carbohydrate? What is the building unit of carbohydrate? Monosaccharide. What's the building unit of fat or lipid? Fatty acid. What's the building unit of protein? Amino acid. What's the building unit of nucleic acid?
Nucleotide.
Nucleotide. Okay? What's the nucleotide? The nucleotide consists of 3 parts. Sugar, phosphate, nitrogenous base. What type of sugar? It depends. Are you talking about the DNA or RNA? Both of them are nucleic acid? Yes. Okay? So phosphate is phosphate. It doesn't change. Sugar, which sugar? If it is DNA, it's deoxyribose. If it is RNA, it's ribose. Okay? Plus the nitrogenous base, which can be AGTC, or instead of the T, In RNA, it's U, and we will see that. If you look at this structure here, this is the sugar, phosphate, nitrogenous base. This is generic. This is nucleotide. Which one? Anything. Any nucleotide. Nitrogen is not going to be different. Phosphate. Sorry, phosphate is not going to be different. Most of the nitrogenous base are not different, just one of them. Sugar is different. From the chemical point of view, you have adenine and guanine, AG. Those are called purines. Okay? And cytosine, thymine, uracil, these are pyrimidines. Now, purines in DNA, RNA, same. No difference. Okay? So, purines, adenine and guanine. DNA, adenine and guanine. RNA, adenine and guanine. How about the pyrimidines? Cytosine in both of them. What's the difference then? The second one. Only one of them. Thymine in DNA is replaced by uracil and RNA. So 3 are the same in both of them. The 2 purines and one of the pyrimidines, which is the cytosine. The only one different, 1 out of the 4, is thymine versus uracil. This is the only one that's different in those two. If you look at this table, I made this table just to summarize it. ATCG versus AUCG. Look at the red. This is the only difference, right? ATCG, AUCG. So the other 3 are the same, right? This is for the nitrogenous base. How about the sugar? DNA, deoxyribose, RNA, ribose. Did you get the difference in the structure? Okay, it's always nitrogenous base, sugar, phosphate. Phosphate, same thing. Sugar, deoxyribose versus ribose. Nitrogenous base, 3 of them are in common, just one different. Thymine versus uracil. Okay? What else is different besides the structure between DNA and RNA? DNA is a double strand, and the D goes with the D. Double strand, like this. This is one strand, RNA. This is DNA, double strand, like this. Here is one strand, here is other strand, they go like this. All right? Folding around each other. So 2 strands, double, the D goes with the D. Double strand. DNA is double strand. RNA is a single strand. Single strand of what? Nucleotide. Okay? So it's a strand of nucleotide. Nucleotide, 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 nucleotide. It's a strand of nucleotide. The DNA is 2 strands of nucleotide. This is the second difference. And obviously the function is different. So see how it looks like? Is this a DNA or RNA? DNA, double, D. DNA, double. Okay? And there is a way that these nitrogenous base, if you have 2 of them, how they are going to fit together. Nitrogenous base is going to fit them together. Which one fits with what? It's not randomly. What?
A goes to T.
Yes.
C goes to G.
Exactly. So the A can fit only with the T. The C can fit only with the G, and always remember it at CG. At CG. The A fits with the T, the C fits with the G. Can you fit the A with the G? No, you can't. It's like this. This is the A and this is the T, they fit like this. Okay? This is the C and this is the G, they fit like this. Can you fit this into this? No. It has to be at CG. RNA can be messenger, which is called M, transfer, which is T, ribosomal, which is R, RNA. This is just summarizing the function that we talked about, the differences in structure and function. ATP is adenosine triphosphate. Adenosine triphosphate. Okay, let's understand this. What's adenosine triphosphate? Okay. Okay. This is adenine. Adenine. One of the nitrogenous base. Okay? ATCG. Adenine. This is one of them. When you add to adenine sugar, you call it adenosine. S-I-N-E. At the end of any word means sugar. So you adenine, you added sugar to it. What do you call it now? Adenosine. Adenosine. I will add phosphate. Adenosine monophosphate. How about this? This is adenosine triphosphate. One more time. What is this? Not adenosine yet. This is just adenine. Adenine. Nitrogenous base adenine. I'm adding sugar. What do you call it now? Adenosine. Adenosine monophosphate. Adenosine. Adenosine. Why are we doing that? Here is why. This is adenosine. If you wanted to add a phosphate, you have to add a glue to stick it together. This glue is energy. You store energy. I wanted to add another. This is adenosine monophosphate, right? Can you add another phosphate to make it adenosine diphosphate? Yes, I can, but you have to store some energy in, like a glue. So put some energy in here, and then I put the phosphate, right? You wanted to add the third one? Yes, give me some energy. Put the energy in here, and add the triphosphate. So this is your way of storing your energy. You stored your energy now. I want to need my energy. I want to use my energy. I need it. Okay. Remove one, release the energy, use it. I need some more. Remove one, release the energy, use it. Okay? Most of the time, it doesn't go like this. Most of the time, it's just you remove one, release the energy, that's it. So ATP, when you remove one phosphate, what do you call it now? ADP, or adenosine diphosphate. Now I'm eating, I'm getting more energy, put the energy back, and return it to ATP. So the storage form is adenosine triphosphate. Remove phosphate, release the energy, and use it. When you get more energy, please give me my energy back. I will put it, add the phosphate again. So it's going to change. It's yours? Any one of those is yours? Okay, so you got this? So it's always changed from ADP to ATP and back from ATP to ADP. The enzyme here is called, I'm almost done, just give me a minute. ATP, when you break it down, the enzyme that work on it is ATPase. Here is another rule. And remember these rules. When you see ASE at the end of any word, it means the enzyme that break down something. ATPase, the enzyme that break down ATP. Okay, lipase, what does it mean? Enzyme that break down lipids. Lipase, lipids. Phosphatase, the enzyme that break down phosphate, and so on. When you add ASE at the end of any word, it means that the enzyme that break down this. So here is the structure of ATP. Okay, this is talking about the turnover. How long does it take for recycling the cells? Each one is different. The liver take 5-6 days. The muscles take like 30 days. Neurons, it takes forever, like 200 days. Fat cells, 15 and 20. Okay, and that's it for this part. For this chapter. This is the end of chapter 2.